The number of people joining going a little slower. Thanks everybody for taking time to be here today, even though the university is closed for snow and ice. I appreciate you taking the time, and I hope everybody is staying safe and warm. It's my pleasure to introduce today's speaker, Dr. Shawn Levy. He is Chief Scientific Officer and Senior Vice President of Applications at Element Biosciences. Shawn leads a multidisciplinary team there supporting sequencing applications development, which includes external partners at places like UGA. Shawn has a deep history in genomics. He received his PhD in biochemistry from Emory University and was a faculty member at Vanderbilt University's Medical Center in the Department of Biomedical Informatics and the Department of Molecular Physiology and Biophysics. He assembled a great genomics core while at Vanderbilt and then did it again, but bigger and better at Hudson Alpha, which subsequently became Discovery Life Science. While a faculty member at Hudson Alpha, Shawn's genomics facility supported more than 5,000 projects from around the world. So literally, it was one of the top 20 core labs that you would find in the US, maybe top 20 in the world. He is an author of 200 peer-reviewed publications with over 45,000 citations. So Shawn's professional journey has included positions in academia, research institutes, and now private industry. I've had the pleasure of attending several of Shawn's seminars over the years, and there is one technical example I still use with proper attribution to this day. So today, Shawn will be telling us about single-cell multiomics and integrated sequencing with instruments from Element Biosciences. Welcome, Shawn.
Thanks, Travis. Thanks for the detailed introduction. It's great to be able to present here. Fortunately, we had a few moments to spend at the University of Georgia yesterday. Would have loved to have been there in person today, but understand the weather, it is what it is. But it's really appreciate everyone's time joining here virtually. I'm joined by my colleague, Chris Higgins, who's the local rep that covers UGA in the Southeast. So between he and I and the rest of the team, we're certainly more than happy to answer questions either at the end of the talk or at any time in the future. So as Travis mentioned, I'm going to discuss some of the unique capabilities that Element has launched. I joined Element after, as Travis described, a little over 20 years in academia. Stated very simply, after being a big customer of genomics for a long, long time, and after my laboratory was sold by Hudson Alpha to a private company, Discovery Life Sciences, it was a great time to take an opportunity to switch to the other side and be able to participate in the launch of a new platform and help guide some of the decisions around that platform. And you'll see, I think, the compendium of those decisions, not only my influence from bringing an academic voice to the company, but as well as a large number of outstanding scientists and technicians that have been at Element since its founding about 7 years ago. What Element has done is really tried to reimagine sequencing, again, trying to build on the building or build on the foundation that was laid in front of it from companies like Illumina and Ion Torrent, Roche, 454, et cetera, to ask the question, how can we advance the technologies forward? And I'll have two primary technical examples today of what we think is some of the more highly differentiated features. I'll touch on the sequencing capabilities of Element, but really wanted to focus on the integrated sequencing, which we call Trinity, as well as the single cell in situ multiomic capabilities that we've recently launched on a platform which is called AVITI 24. It's essentially an upgraded version of the original sequencer, which means so we don't have two platforms on the market. We really have one. You can either have it in the upgraded format, which is 24, or the standard sequencing format, which is AVITI. So they are essentially the same machine. One just essentially has upgraded software and substantially upgraded compute because of the amount of computing horsepower that's required to run the assays that you'll see in a few moments. But otherwise, we really wanted to. What you'll find in this is a platform now that has significant capabilities in both sequencing, really finally reinventing targeted sequencing. Again, you'll see that data, and then I'll end the talk with a walk through the multiomic capabilities and some example data sets in some very basic drug screens, et cetera, with the idea that just showing that it really works, and what those capabilities are. And we'll offer a little bit of insights and a couple of things that are coming this year in 2025. So, one second. There we go. So none of this would have been possible without the contributions of so many outstanding people at Element. This is a company photo, and I have the privilege today of really representing the work of all the people you see here. So it's been a great team to work with over the last 3 or so years, and hopefully you'll see the output of a lot of these efforts today in the talk. So very quickly, the platform that we're describing, I'm just closing a couple of windows. So this really, I'm introducing the platform 24 that I mentioned a moment ago. On the left-hand side of the slide, you see the specifications around sequencing. So this is a product name that we call Cloudbreak. That's essentially the name of the sequencing product. I won't go through all of these things, but you can kind of read them on the slide. We tried to have the highest quality sequencing, flexible output per run, as well as a very high value in terms of cost per million reads or cost per gigabase of sequencing. On the right-hand side of the slide, you see the specifications for the Teton. This is the CytoProfiling that you'll see data at the end of the talk. So these specifications will make a little bit more sense as we go on, but wanted to highlight two key things. One is using both sides of the instrument. It's a 2 flow cell instrument, 2 independently controlled sides, an A and a B side. When those are used simultaneously, we can generate you can profile about 2 million cells at the same time. This is across 20 centimeters squared of area per run. Runtime is 24 hours. And again, you'll see the data in a moment that we're actually simultaneously detecting RNA, protein, as well as morphological markers via cell paint. So all this will make a bit more sense as we walk through the data. So Element, how this is possible, and this will kind of integrate, this will give you an idea of how we've integrated the sequencing capability into some of these other capabilities, but wanted to at least show you what the foundational technologies or the new features that Element brought to the field to enable this type of instrument to be produced and this type of data to come off of that instrument. First started with a very flexible surface chemistry shown on the upper left-hand side of the slide. This allows us to essentially this is a non-patterned flow cell with a very flexible surface chemistry. That surface chemistry can be easily changed, modified, and we can even use multiple surface chemistries on the instrument for different purposes. So this allows us to have things that you see highlighted, whether it be cell capture or cell culture, protein detection, targeted capture that you'll see today, or just straight sequencing. The Element spent a long time developing a very robust, very simple surface chemistry that has the ability to be very, very dynamic. The next feature is a low error amplification. We use a modified version of rolling circle to amplify nucleic acid on the flow cell. The reason that's important is we cannot generate copies of copies. So if there is an error during the amplification, that error does not propagate. This is one of the key features in terms of the sequencing quality and sequencing accuracy on the platform. So we were able to generate multiple copies of a library insert, but those multiple copies of that library insert cannot propagate error when invariably those happen due to just the normal process or the molecular biology process of polymerases. They're not perfect. They can be pretty close to perfect. That we took an opportunity to make sure that those errors could not propagate through to the sequencing. Finally, and most significantly, is the ABC, the AVITI-based chemistry, or sometimes referred to as AVITI-based chemistry. So I'll use the acronym ABC. You'll see that in some of our papers and things like that. This is a very unique sequencing chemistry. I may have one more slide that details this in a slightly different cartoon, but what I want you to appreciate from that cartoon on the upper right-hand side is the nucleotides are separate from the fluorescent tag that identifies the nucleotides. And think of an octopus is probably the best analogy or the best one I've come up with, where the fluorescent molecules are held in the body of the octopus, and then the octopus has multiple tentacles, and at the end of the tentacles is where the nucleotides interact with the DNA. So let me see if I can pull up a pointer real quick. So if we focus here, these 3 red cartoon pieces here, think of these as a sequencing primer, and then you have 4 different polymerase molecules that are measuring what base would come next to allow that complementary base to interact. At the end of the tentacles is a nucleotide. That nucleotide then interacts with the polymerase. And so essentially you have a multivalent interaction, meaning that you have to have that for a given avidite to interact, it interacts with multiple copies of the insert at the same time. So this allows, again, another factor of, call it fidelity in the process, or error, or presents another opportunity to have a very low error, very high quality or high accuracy sequencing call. Because if one of these is an error, say, either phasing or pre-phasing error, you can still have three interactions that are correct and then measure this fluorescent molecule. What this allows us to do is we now no longer have a requirement for cleaving off a fluorescent molecule. We don't actually incorporate the nucleotide at this stage. This is just sensed, and then we wash this away with a buffer exchange, and then the actual nucleotide incorporation is a natural nucleotide is incorporated. So this means there's no scarring. We have a very, very low phasing and pre-phasing problem. We have a very low introduction of non-natural nucleotides in the process, and this all together allows the sequencing process to be very, very high fidelity. The other key thing is this is efficient enough because of this multivalent interaction and the way these fluorescent molecules are incorporated, that the kinetics of this reaction, and let me see if I happen to have that slide next, which I don't. So we'll just maybe summarize it in, these are the sequencing steps. So here's that same figure blown up a little bit bigger. So we bind the avidite, wash, then detect the base with the imaging system, remove the avidite with the sequencing, and then we step with this natural reversibly terminated nucleotide. So it's just a 3' block, very standard chemistry, that we remove the block and do this whole cycle all over again. This QR code is a link to a Nature Biotechnology paper that we published early in the company's history that describes the avidite sequencing process and some of the technology behind it. And I won't go through each of these bullet points, but you can read them, that these are some of the key features that the sequencing allows. This is a reminder that I tell you that it is a complete 4-color sequencing chemistry, and that you can, of course, have multiple avidites are interacting with that amplified polyny at each of these cases. So taken together, this is how the cycle sequencing works. It is very unique from other sequencing by synthesis chemistries in that we've separated the avidite step, the binding of the nucleotide, the fluorescent nucleotide, from the stepping, the actual extension, by using 2 different nucleotides for that, one of them being in this avidite construct and the other being a natural nucleotide. So that's the basic of how the sequencing works. As I mentioned, much more detail in the Nature Biotechnology paper. So Element has been quite busy since launching the platform in 2022. This is kind of our product portfolio. So we've introduced the cloud break chemistry, a few different options as far as kit output and individual lane addressability. All the flow cells are 2 lanes. They can be individually addressed. That individually addressable lanes is done just with a software change. So you don't actually have to physically interact with the flow cell differently, say off instrument. You simply load either 1 or 2 libraries into the cartridge, and then the instrument takes care of the rest. We can go all the way out to 600 cycles of sequencing in the same run. So we launched a 2x300 capability in 2024. Our freestyle chemistry allows linear loading, as well as compatibility with Illumina formatted, the P5, P7 formatted libraries, which have become so ubiquitous from different vendors across the field. So whether it is an Element formatted library, which we call Elevate, or an Illumina formatted library with a standard P5, P7 adapter, both of those libraries are natively compatible with the sequencer via our freestyle chemistry. We also launched an UltraQ, which is a very high quality capability. And then finally, what we'll focus on today is the AVITI 24 platform, as well as our Trinity on flow cell enrichment. But any questions about the product portfolio, et cetera, there's information on the website, or Chris or I will be happy to answer any of those questions. We haven't forgotten about the software. I won't spend much time on this other than to indicate that Element Bio Cloud is a software solution that allows remote run monitoring, remote run setup, whether it be on your PC or a mobile device, and that we've integrated the instrument for a very convenient connection to things like AWS, Google Cloud, really your favorite cloud provider platform, with our ability to manage the fleet, to plan runs, to manage cloud storage connection, and even monitor run progress. So this is all as an integrated feature. We didn't want to develop a full end-to-end analysis portfolio and force customers into our ecosystem. So what we did instead is created an ecosystem to allow very convenient data access and data connection to providers of choice, some of the ones I mentioned here, as well as a long list of others. So this way, we've just enabled easy data transfer for whichever methodologies, programs, or resources are most appropriate for a given experimental design. So freestyle, as I mentioned here, this allows us to perform the circularization of any library type without a separate conversion or circularization step, meaning everything happens on the instrument. It's all native to the platform. Very unique feature. I think this is the first platform that has allowed that compatibility with multiple library types without using a separate conversion step. So if we look at this in terms of bringing everything together, like how well does the sequencing work and where do we see some differentiation points? This is a bioRxiv paper. It's currently in peer review that was from Andrew Carroll's group at Google Health. And this is the title of the paper. Here's a DOI link to that for anyone interested. And essentially what we asked a couple of questions, which was simply, we did human genome sequencing using a reference sample where there's a known truth set, in this case, HG003. And then we performed the same bioinformatics pipeline was used for this. Essentially, the Google bioinformatics pipeline was used for this. And then we looked at combined SNP and indel precision versus SNP and indel recall, and then did a couple of different experiments. One was standard element sequencing, which is the solid blue line, versus Illumina at 4 different coverage levels, which are represented by the shapes, 20x, 30, 40, or 50x. And then we also introduced some element long insert data. So this is Element's ability to use sequencing insert sizes that are much larger. In this case, about 1.3 to 1.5 kb in terms of insert length versus, say, a traditional library type, which may have 500 or 600 base pair inserts. And so the key thing is the sequencing read lengths are unchanged. These are all paired 150 base pair read lengths. What the long insert does is essentially just separate the space between those reads about double what a standard insert is. And so you can see this fairly significant increase in, particularly for indels, which is, I know that's combined here, but you see this shift really at all coverage levels, particularly at as these climb from the 20x to 30x to 40x. We didn't have 50x long insert data, but you see, and the reason we didn't run that is you can see the star and the square are very, very close here on the normal insert. So we knew it wasn't going to be dramatically differentiated here. But what this means is for all sequencing technologies, no matter how accurate the base calls are, if the inserts are close together, you get less unique mappings in the human genome, and those mismaps, particularly in when you're using a single non-pangenome reference. A lot of the error that's driven in the final sequencing results is actually alignment and variant calling error. And so we've recognized that if we just shift those inserts to a longer insert length, those inserts would then allow us to more accurately map. And then this paper goes through that in a lot more detail. So just showing, just even on the straight sequencing side, there are some experimental designs and opportunities where the element chemistry and the capabilities on the platform do become quite differentiating. So what we really started off with was some of this best-in-class sequencing, both from a quality as well as affordability. So the affordability, this $200 genome on a benchtop, that's a volume-based program that if you're running the equivalent of about 1,500 or 1,500 or more genomes per year, you can access pricing in this ballpark. And then a very, very high reliability, a mean time between failure of greater than 150 days. So now we wanted to start simplifying those workflow, onboard library prep, multiomic profiling, and just continuing to scale the platform so we can do more experiments in less time. So recognizing that these features are kind of the table stakes, and now how can we continue to bring additional value to users of the platform? And then that's what we'll focus on for the rest of the today. So new innovations in exome and targeted sequencing is what I'll talk about next, and I'll kind of walk through this quite quickly. And this is the Element Trinity product. For those of you familiar with targeted sequencing, I think conceptually you'll recognize this as fairly simple. So the traditional hybrid selection process, which has really been around since the early 2000s, in some forms, and then probably really popularized with some seminal work out of the Broad Institute in 2007, 2008, that became the foundation for what Agilent launched as their SureSelect technology, which was an RNA-based bait, which then rapidly evolved into DNA-based baits from companies like IDT and Twist. And now we have, as well as companies like Nimblegen, which was acquired by Roche. So we ended up with a very robust ecosystem of content, whether it be exome or targeted panels from a variety of different vendors, but they all shared commonality in the workflow, and that's summarized here. And that commonality was the production of a DNA library, biotinylated target probes that were hybridized together first on microarrays in the early days of Nimblegen, then became in solution with the SureSelect technology, again, that publication in the mid 2000s or so. That probe hybridization, typically 4 to 16 hours. And at the end of the hybridization, you then bound the streptavidin or you bound the biotinylated probes to a streptavidin bead, a magnetic bead, which then allowed you to capture the sequences of interest, went through a number of wash steps, followed by a post-hybridization PCR, library QC, and then loading onto sequencing cartridge. So this workflow is still robustly used today for nearly all targeted sequencing that's not PCR-based. And this aspect of the technology, starting from the bead binding through the washing, is fairly technical to maintain high degrees of consistency and high degrees of performance. Temperature control is very important. Avoiding evaporation is important. And then, of course, you are subject to the second round of PCR to make sure you have enough library to then sequence. In a nutshell, what Trinity did was eliminated essentially all of these steps. So now this is truly the workflow where you still do the same library preparation, still use your favorite bait molecules here, the biotinylated target probes, perform that hybridization, but now you can reduce that time to as short as 1 hour or still maintain the flexibility of using that out to an overnight hyb if that works in a workflow. But at the end of the hybridization, you literally just load the cartridge and then goes on to sequencing. Now, to be clear, it's not just that these steps are performed on the instrument. It's foundationally a different technology. We actually capture, this step is captured on the flow cell. We have no need for this step any longer. And then the library QC is essentially taken care of on this original QC of the library. So uniquely, this means the representation in the sequencing here is a much closer representation of this original library by avoiding this PCR, and you'll see some of those effects. So and then we also are working on a Trinity Plus workflow where this hybridization step actually occurs on the cartridge. We haven't given any guidance yet as to exactly when this will be released, but mainly because with this 1 hour hybridization workflow, this is a really robust, and it has some features of flexibility that are kind of lost in this, but there are some plans for applications that will launch this a bit later. So some of the highlights of the features and differentiators, we get a higher complexity library output. You'll see that in a moment in some data. Dramatically simplified workflow, meaning that you just don't need any of these technical steps. If you can prepare a library, you can absolutely go all the way through to targeted sequencing in a flexible manner. We see lower error rates and variant calling primarily through the avoidance of this PCR step, much shorter workflow. And the workflow outperforms the standard protocols for all the partners we've tested so far. We formally launched this with a product with IDT and another product with Twist, and we'll have more partners launched in the future. So in summary, here's our on-target performance shown on the right-hand side of the slide for exome. In solution is in the light green. Trinity is in the darker green. So on-target is in the high 80s to low 90s. That's for an exome. And then here versus a custom myeloid panel, you can see our target on-target rate is a bit higher than that. On-target coverage, call it very comparable, both for exome versus myeloid panel. And then here's our total assays time shown on the left-hand side of the slide. Trinity Fast Hyb, total time is around 5 hours. That's using a 1 hour Hyb time, about 4 hours for the library preparation. Standard Hyb takes that out to about 20 hours, again, about a 16-hour Hyb. And then the on-market Fast Hyb and on-market Standard Hybs are kind of shown in the graphs here. So you can see we've just taken a lot of the time out of the workflow overall. So how this works, so now, because we're using the flow cell as the capture panel, you have to kind of think of things sort of from the flow cell backwards. And what makes it unique is if you start thinking, well, because we are seeding the flow cell with the entire population of nucleic acid that's in the mixture, that means your input has to be representative of what your expected output would be. And this circle here is a good example. So think of this as if you start with exome, let's say the content of the exome, call it 30 megabases in size. And then you want to contrast that with the size of, say, one model panel here versus model panel number 2, it's the size here. So call it maybe model panel 1 is 2% of an exome size. Model panel 2 might be 5 or 10% of an exome size. The reason that's important, if you think of the idea of how many molecules are necessary to make 1 billion sequencing reads on a flow cell, the exome would, of course, require maybe 1/10 of the input of a small panel. Because when you think of that high specificity of capture, now when you load everything on the flow cell, you need enough material of the material that you want to sequence to actually seed those billion reads. And so recognizing that this is a little bit of a different way of thinking of it, we've generated these types of graphs so that you can relate a specific microgram of molecules needed to generate 800 million to a billion sequencing reads to the panel length. And then so this is different size panels. And you can see there's a pretty linear relationship between how much material is necessary for 800 million reads versus panel length. Kind of maybe avoiding having to read these very small numbers on the screen, I'll distill it down to say that generally speaking, it's 125 nanograms to 250 nanograms of each input library per sample that goes on the flow cell, whether it's an exome or a panel. What this means, though, is that, so you say, well, if I'm running a really small panel, I'm putting 96 or 192 samples on a flow cell, that's a whole lot of input material into the Hive. And that's true. And we have protocols to actually balance that to make that convenient. So we're very fortunate that this performance has been linear. What I would just caution everyone to say is when you first glance at that and relate it back to a traditional exome protocol, you'll think, wow, that is a lot of DNA going into the panel, but it works fine. It's kind of once you kind of get around this is a little bit of a different way of thinking. It becomes very, very comfortable. So when we look at the assay performance over even in low quality samples, in this case, we looked at 8 blood versus 16 FFPE samples. Those FFPE samples were 8 tumor biopsies and 8 resection samples that were matched to the original 8 blood. We used 50 nanogram inputs of DNA into the library prep workflow. And then for PCR, for Trinity, we did 10 cycles of pre-hybridization PCR, and in solution, we used 7. This was, again, just to boost this yield a little bit. And you can see when we look at on the Trinity workflow, which is the darker purple versus in solution, we saw over most of the samples, maybe one outlier here, a consistently lower percent duplication rate. This has been consistent on all sample types. And then maybe most importantly, we see a higher, notice Trinity here in purple, a higher mean target coverage per sample versus number of reads, meaning for the same number of reads, we saw higher effective coverage on the Trinity samples versus the same exact input libraries into the in solution. So what this means is we could actually use about 10% fewer pass filter reads to achieve a certain coverage, mean target coverage per sample. So the sequencing was a bit more efficient, and a lot of that's driven by this lower duplication rate. Some of it's driven by just a higher specificity in the capture. We also see that at the same read depth, we see higher diversity in the library. The reason for this is the capture efficiency is a bit higher on the flow cell compared to the in solution bead preparation. And again, avoiding that PCR where you don't have any jackpotting of certain features of interest. We generally see higher complexity libraries. Notice Trinity versus in solution for some of these, whether it be the blood samples versus some of the biopsy or resection samples. So overall, you can kind of see from these graphs. So overall, we see in these graphs a bit higher performance metrics for a number of these parameters. And you can kind of see here total reads per sample, the on-target rates for in solution versus Trinity and some of the fold 80 coverages. So most of the other metrics look quite similar, except for this difference here in the duplication rate. And then notice here the library size. A pretty significant difference in expected library size, again, measuring that higher complexity in the library. What this translates to is an ability to detect variants at lower frequencies with statistically significant performance. So I've been talking mainly about panels or exomes. We've also looked at things like RNA capture, and this is using an RNA library with the Twist RNA exome kit across 24 samples. Just show you that there's a high degree of strand specificity that you can see here in this lower graph, and then in the upper graph, very high degree of on target rate. And most importantly, across these 24 samples, you just see very consistent performance across the samples. This is mainly illustrating that this same workflow works with a wide diversity of capture probes, panel designs, and sample types. So we also, if we look at a very small custom myeloid panel, observed allele frequency was exactly as expected. In this case, this was using a 1-hour hybridization workflow with a quite small oncology panel, only 7,900 probes, not genes, probes. So we use 10 samples with some controls, and then you can achieve very high coverage, in this case, 3,800 to almost 10,000 fold coverage. And 22 out of 22 cancer mutations that were expected in this control sample were detected, and there was a high correlation with the reference, as you see here. So this, again, just illustrating that even if you go to a very, very small panel, the workflow still remains very robust. The last thing I'll mention on this is you can mix panels together to take advantage of sequencing performance. In this case, we were mixing an exome with an IDT pan-cancer panel, and what we were aiming for is 80% of the reads to exome, 20% of the reads to the pan-cancer, and you can see we actually achieved 77 and 23 respectively, so good performance here. High quality, good number of assigned pass filter reads, almost 1 billion reads here on the flow cell, numbers that I mentioned right here, and then very high on-target rates for each one, 93% for the exome and 84% for the cancer panel. So again, just showing that you can still retain a lot of flexibility on the platform by sharing the sequencing reads of a flow cell with the Trinity workflow, even on multiple panels, even if those panels are significantly different in size. So the last thing is, because we don't require PCR, we can do a PCR-free workflow. And just briefly, I'll show you, here is traditional whole exome sequencing versus Trinity, and this is our total false positive and false negative reads here. What you're looking at is error. So if we look at our false positive plus false negative errors here for any of the passing variants, this is the total number, and then we can be looking at false positive and false negative in SNPs in these two colors, and then false positive and false negative indels in the green and red. And essentially what you're seeing is you can drop those numbers by, call it roughly 40% in this PCR-free workflow versus traditional. We also see that it's really dramatically different in the indel F1 rates versus the SNP F1 rates. I think both are improved, but you really see this difference here, and that probably comes as no surprise. You see that in even whole genome sequencing, that with PCR-free workflows, we see a better improvement in performance with indels versus SNPs for the most part. So how this looks if we kind of cherry pick a couple of regions. Here's an IGV snapshot of traditional exome versus the PCR-free. And by a couple, there's a couple of mechanisms here. Some of it is not biasing to shorter inserts with that secondary PCR. Some of it is just not having a GC bias into the PCR workflow. But the bottom line is you can kind of see some of these regions of the genome are covered a bit more robustly in the PCR-free versus the traditional exome. And in some cases where you're actually calling, these variants would have been missed and counted as false negatives in the traditional exome, but actually covered pretty well in the PCR-free. Now, we fully recognize this is not a workflow that would be appropriate for, say, every experimental design in every sample. I'm showing this example that for conditions when it is advantageous, it's available, and it's just an inbuilt feature of the platform that can be used when when necessary. So that kind of ends the Trinity portion, and I'm going to really entirely shift gears now to the integrated single-cell multiomics and spend the last 20 or so minutes on this section. And I'll hopefully show this is, again, pretty significant change, because really what we're talking about now is on this, stepping away from sequencing on the platform, but instead using the same foundational technologies, surface chemistry, the ABC sequencing chemistry, but turn them in a very unique way to I think propel the type of data generation that's going to be necessary to drive us into the next inflection point in understanding of complex biology and multicellular organisms. I think everyone recognizes in the genomic era has kind of ushered in the idea that molecular data across multiple modalities, whether it's the idea of protein phosphorylation for changing protein activity to protein measurements overall, to then incorporating transcriptomics and then DNA sequences, that all of these modalities play important roles in looking at biology. And we've seen technologies come out in proteomics and transcriptomics and even genomics, at the genome scale, that have been very, very revealing. But it has not been very long since we've been able to start orienting these methodologies or these types of detection with actual cellular morphology or tissue morphology or functional readout. And that, in fact, has been a significant opportunity for technologies in the future. So I wanted to maybe mention two things to orient things, and also what I found is some very interesting reading. So there was a publication that came out just before the holidays in Cell from Dr. Quake at at Stanford, and he also is the president of the Chan Zuckerberg Institute. And it's a great short commentary, provides a really elegant reminder that our understanding of information flow, essentially, our information flow in biology remains very limited. So one of the examples that Dr. Quake uses, which I found pretty compelling, is even with all of our knowledge and all of the genomes that we've sequenced, we still have an inability to start with a genome sequence and then predict only from that genome sequence how many cells, how many cell types, and what is the pattern of cell differentiation or cellular development that is encoded by that genome. We still lack that connection of genome information to, call it final biological information. And what he proposes in this, and again, it's a very nice commentary. I would encourage you to read it. It's about 2 pages long or so. Again, it's really, really well written. And I think it kind of provides a stage for what we need to think about for maybe propelling us into the next decade or so. And this all comes down to kind of how information flows in biology. Separately, there's a great paper that came out in May 2023. This has now been cited I think about 400 times now, from Christina Theodoros, who's now at the Gladstone. This was some work when she was a postdoc at Harvard with her colleagues from Harvard, shown here in the author list. And this presents the model GeneFormer. It's a pre-trained AI model on about 30 million single-cell transcriptomes, is what was shown in this paper. And that enables some context-specific predictions and settings with limited data in network biology, basically saying that this was an ability to kind of predict some of that information flow in biology using single-cell transcriptomics. Since this initial publication, GeneFormer has been further trained with data from the Chan Zuckerberg Cell by Gene Census. This is an effort across the CZI Institute to continue to add more tens of millions of additional single-cell transcriptomes to it. So the reason I mention these two things is, one is this is a great commentary on kind of maybe some ideas of how we can look at opportunities in the future, and then also the recognition that AI-based technologies and the ability to start integrating these very, very large data sets is in front of us, but we still need technologies that allow us to make observations in this multimodal fashion to really take advantage of what the computational power and capabilities in AI will bring us. So stated a little bit differently, today we have to use multiple workflows and tools to assay biology. These are just some examples in RNA, morphology, and protein. But is there an opportunity to kind of look at these things simultaneously? And what Element has developed that is possible on its 24 system, and I should make a clear note, everything you'll see in the next several slides is all captured on the AVITI instrument. And if there's anything else that was done like as a companion assay, I'll call that out. But any of the imaging you see, including this slide here, this was all captured on the AVITI 24 instrument. So remember, this is the same platform, same instrument, that if you can use it on Monday as a sequencer, you can use it on Tuesday as a multiomic instrument, all in the same feature. And in fact, you can indeed run side A as a sequencing workflow and side B as a multiomics workflow at the same time. So you could actually use these, both on Monday. But this is showing 6 targets in cell morphology, where this is essentially Element's version of cell paint. So cell membrane, nucleus, actin, endoplasmic reticulum, mitochondria, and Golgi can all be captured. And then we can also simultaneously, just captured in a slightly different data collection, but on the same cells, look at RNA detection. 350 targets is what's shown here. So each of these colors represents a colony that's detected. And then we can also do protein detection, 50 plus surface, intracellular, and even phosphorylated proteins that can be done. So essentially in the same experiment, you can look at morphology, targeted RNA, and then targeted protein. At launch, we've launched a MAP kinase cell cycle and apoptosis panel, and coming soon in 2025 is an immunology, neuro, and even the ability to do custom panels. So I'll be showing you some data on these early launch panels, but know that more is coming. So this is a screenshot from the software, and you'll kind of see this cycle through in the video, choosing different proteins, in this case proteins as well as transcripts, to light up in the background of the cell membrane, nucleus, and actin, the green, blue, and red that's already turned on. So this is Element software. This is the software that comes with the AVITI 24 system, showing how you can interact with that. And you'll notice that you can also turn on the cell segmentation. You can also turn on then the nuclear boundaries. So there's quite a bit of ability to interact with this. We've made the data formats as open as possible, and definitely support the ability to use things like Seurat and other models. So you can export this in JSON-like format, so that again, you're not locked into any one analysis platform. We know that this is going to be a very, very active area of development. So as I mentioned, we're launching with a couple of different panels here. The 6 morphology markers that are shown here. And this idea is that you can really carefully look at MAP kinase and cell cycle and apoptosis pathways, some of those foundational pathways in many, many areas of cell biology. Stated a little differently, these panels actually make for very good developmental biology, and also even cancer profiling panels right out of the gate, even though we've kind of titled them as a MAP kinase, cell cycle, and apoptosis pathways. They're very, very relevant, and they contain many of the things you see here: cell cycle regulators, pre-apoptotic factors, anti-apoptotic factors, stress response, and DNA replication and transcription factors are all pretty well represented. So how this works is we have a flow cell that looks like this. This is the 12-well version. You see it's a 6x2 format. These are spaced in SBS format, or SBS dimensions, so you can use multi-channel pipetting. You can culture or deposit cells directly on the flow cell. This flow cell here, this cover comes off, so it's openly accessible. You can put up about 50,000 cells per well in the 12 well. We have an open well design. Kind of think of it like this here, which can fit up to about 1 million cells if you're using it in a single well design. So there's a sample, you do the sample preparation, which is a wash, fix, and permeabilization of the cells. We have both adherent cell and suspension cell protocols. So after this 1 hour fixation and permeabilization step, you simply load it onto the sequencer with the appropriate cartridge and then collect the data about 24 hours later. So we've tried to make this as seamless and as simple as possible. We should note that if this is done in a laboratory at a different location than the sequencer, once the cells are fixed, they can be shipped or transferred, and then the permeabilization steps are performed. Flow cell is assembled and then run. So there is that level of flexibility. As I mentioned, open data format, to allow off-instrument downstream analysis. We allow easy integration with many of the community tools you see here. And then these are some of the different data steps that we've incorporated into either using these tools or into the Element software: cell segmentation, differential expression, morphology, pathway analysis, clustering, network analysis, et cetera. So again, we've tried to build this out as an ecosystem with as comprehensive analysis as possible. So here I just show you some examples of kind of applying this now in an end-to-end format, looking at some drug response with the CytoProfiling. So this is again, very simple experiment on well-known cell lines and looking at well-known pathways. So we used a single dual-sided run with 2 treatments. We used 12 time points total, essentially 24 hours of data and up to 50,000 cells per well. So in this, again, this simple experiment, 1.2 million cells total. On side A, we ran a TNF alpha treatment across those 12 conditions. And on side B, we used an EGFR kinase inhibitor treatment. So these are the pathways that are typically affected. In both of those cases, again, we wanted to just try to recapitulate well-known biology. So here you're looking at drug-induced changes to RNA, protein, phosphoprotein, and morphology. So what you'll see here is just kind of some vignettes across this data. These are the time points that were used: 0, 5, 10, 30, 60, and 120 minutes for the kinase inhibitor. And you can see and you just kind of scan across for each of the proteins that are shown. In this case, protein HSP27 versus phospho HSP27. You can see here around 30 minutes or so, you see a peak, falls off. If we look at RNA phos, same thing. You see this in here. And then if we look at morphology, you can look through these morphology changes. This is just one small window across the 50,000 cells or so per entity. And then if we look at this now in a UMAP type format, we can start to see some of the interesting biology. TNF alpha here on the left, EGR kinase inhibitor on the right. In one case, looking at wild type versus different concentrations of drug, or in the TNF alpha case, looking at different time points. And then you can also, if we cluster by cell cycle state, you can very robustly see where the drug dose response is relative to cell cycle position. And again, this is just some small vignettes just to show that walking you through that the fact that we've introduced this technology and that technology produces recapitulate some of these known biology insights. But where it becomes interesting is when you start asking, if we look at well, what happens if you are trying to really understand that drug response? Does looking across these modalities really improve resolution? And the answer is, generally yes. So if we look at both TNF alpha treatment shows a strong protein and RNA response. And so if we look at the single cell classification error, if we use that only a morphology marker, we see it, we get about 70% right. But if we add in, and if we look at only protein or only RNA, you can see we perform a bit better, meaning the signature of the time point of the TNF alpha for RNA and protein is a bit more robust than looking at only morphology markers. But if we combine them all together, this is where you start really seeing some of that more revealing or that more accuracy in this, again, well-known example. And so we think the corollary to this is that if by adding these multimodal together allows you to predict known biology with ever increasing accuracy, it also means that for unknown biology, you're going to be measuring data sets that are absolutely more predictive or more indicative of that biology. And we can do the same thing in this tyrosine kinase inhibitor treatment with a slightly different performance in terms of protein versus RNA, but the same trend. Protein here, RNA. In this case, protein had the highest error. RNA was better. Morphology was actually more predictive if you notice this contrast here. But then when you, again, put them all together, you have your highest performing, your highest accuracy point. So some of the basic performance things, because I know some of the questions that you may be wondering is, well, how well does this correlate to other technologies? What is the sensitivity? What is the reproducibility between runs? And we've seen them very high. So as far as I'll talk more about this transcripts per cell in a couple of minutes, but just wanted to show we see this very nice distribution of the ability to detect significant numbers of transcripts per cell. So showing that we have a pretty good dynamic range when we're looking at the transcripts. Reproducibility between runs and between wells is shown here. So this is if you just run the same experiment twice, again, between different instrument runs, or the same experiment twice by repeating things in between wells in the same 12-well format. Again, very highly reproducible here. And then strong sensitivity as well as strong resolution, just shown in the heat maps here. If we look at the number of cells versus single cell RNA copy numbers, you can see as we get, once you get down to say on we're only observing single cells with very low copy number, we see some error. But once you get to call it single cell RNA copy numbers of about 3 copies per cell and above, and then you're observing say 100 cells or more, that's when we see some very, very strong correlation or very high sensitivity and good resolution. So we're probably in that couple copies transcripts per cell. As long as you're observing multiple cells, you have a good way of detecting that. If you're observing very, very small numbers of cells, well, then that RNA copy number needs to be a bit higher to robustly detect it, which probably comes as no surprise. With orthogonal methods, if we look at RNA-seq, bulk RNA-seq versus single-cell RNA-seq versus FISH, you can see we can kind of correlate quite well with all of those. Not perfectly, but that has to do with some fundamental differences in the measurement of the technology. But you can see a very, very clear trend in the same direction for each of these technologies, maybe with the best correlation with FISH in the transcripts per cell. So if we ask the same thing with proteins instead of transcripts, we use immunofluorescence detection to look at that correlation, and same good benchmarking versus the immunofluorescence as you see here, IF fold change versus the AVITI predicted fold change. A little bit of a different in terms of scale, so there is that calibration things we're looking at. But here's if we look at an immunofluorescence image here versus an AVITI 24, and then do the merged overlay, you can see that they overlay sort of at the gross level pretty well. So lastly, wanted to mention kind of a little bit of a vignette of what's coming in 2025. And this is where that transcripts per cell gets a little bit interesting. And that is, so all those foundational chemistry improvements that I mentioned, the surface chemistry, the ability to do the sequencing, since we are using cycles of sequencing to directly detect the probes that are used for the targeted transcripts and the targeted protein detection. So there's also the opportunity to ask a different question, which is, well, instead of sequencing a probe in the cells in an in situ manner, which is all of the data that you've seen so far, can we instead directly sequence a transcript in the cells in an in situ manner? And this is the data you're seeing here. So the answer is yes. We can do it in a targeted way that you see here, and you kind of see this cycling through in 100 cycles, or we can look at an untargeted way. What you're seeing here is looking at, and the reason, see how you can see these colors are all coming up the same color. That's because it's in a targeted sequencing manner. And in this case, You see all the different colors that kind of the rain, kind of sort of a rainbow view because this is an untargeted sequencing. Both 100 cycles. This is actually looking at GAP-DH. This is looking at, of course, some of the more highly expressed transcripts. Now, you may be wondering, and I'm not sure how well the resolution is on everybody's screen, but you can kind of see, where there is, you can barely make out. I'm kind of tracing the outside of the cell membrane here for this particular cell, and in this particular cell here. The reason you're seeing some of these things appearing, what it may appear like on the fringes of the cell is this is not corrected for any Z-stack error as to how these images were merged, and that's why you're seeing a little bit of this. So remember, there's a little bit of a Z-stack correction that would have to take place. But if we then bring this together and compare it to a single cell RNA-seq experiment where we looked at 6,500 cells, and in the single cell data simply ordered the transcripts based on most abundant to least abundant. And you see the transcript list here, again, by mean counts. And then we did the untargeted 3' single-cell RNA-seq on AVITI-24 with 60,000 cells in a well, and then simply ordered that data based on the same transcript list. And you can see, we see this correlation here. So not perfect. Maybe we're seeing a few transcripts detected pretty robustly that were missed in the single cell. Then some of these, a little bit of variability in terms of the spikes of coverage here. Some of this is just due to differences in the technology. We're generating about 400 counts per cell, and you can see this is a screen capture of the cells of all of the pollinase shown at the same time. And there's thousands of RNA counts detected per cell over the multiple sequencing rounds. So if some of you may be thinking, if you look at, if we looked at this here, I actually did, it's not showing on what we're detecting about over 1,000 cells, about 3,000 counts. One of the tricks we've implemented into this is actually multiple rounds of sequencing. So we can actually take an important opportunity to differentiate sort of optical resolution versus physical resolution. This is a key capability that will allow Element to continue to advance this platform to higher and higher plex and higher resolution within the cell or within the cell bodies. So there's really a lot of opportunity to move this into new directions and move this into higher and higher plexity and the ability to look at different Z levels on the cells, et cetera. So we're just now scratching the surface of a lot of these capabilities. We're really excited that we can bring this type of biology. So what this now means is, throughout 2025, we'll release capabilities that allow you to go from a targeted RNA detection, targeted protein detection, to now more untargeted, at least RNA detection in this sense, where you can now start asking questions that, I hesitate to call this total transcriptome, because again, obviously we're kind of looking at in this data, looking at the top transcripts. But you at least have the ability now to have an opportunity to look in a targeted way as well as an unbiased, untargeted way, and therefore generating much more flexibility in the overall experimental designs that can be supported. So just as a reminder, this is all happening on exactly the same platform, on the AVITI-24 platform. So very, very high quality, very cost-effective sequencing, with the ability to do this type of cytoprofiling. And then most importantly, we've tried to keep the workflows as robust and as flexible as possible so that a single instrument can be located in a core facility, can be located in a lab, and then allow it to be shared, say, across a campus, where you don't have to do, the sequencing. Obviously, libraries can be prepared in one library, sequenced in another, but our Trinity targeted sequencing, same thing. You can perform a hybridization step in one lab. We have features in the protocol that make that exceptionally stable to allow it to be saved for hours or even days before it goes on the sequencer post-Hive. And then same thing, this Teton cytoprofiling, you can fix the cells on the slide, and then you have a period of time before they have to go on the sequencer to really make sure that this is quite robust. So I'll end there. I'm happy to open it up for the last few minutes for any questions or comments. And again, really appreciate everybody's time today. As I said, if we run out of time, we're more than happy to answer questions in the future. Contact either Chris or I. So thanks a lot. And if anyone has any questions you don't want to ask in person, if you want to throw it in the chat, that's great. We probably have maybe 4 or 5 minutes left before we're at the halfway through the hour.
I put one in there just because I'm curious about it. Oh, and we got another from Oshri.
Yeah. So, Chris asked, do we envision supporting custom panels for Trinity PCR free? Absolutely. There's no, the kind of the library prep is independent of the panels. So can you use tissue on the slide? Great question. One we get often. I'll answer that by saying there's no technical reason why we can't, put other biomaterial on the slide than cells. We've really felt it important to focus on cells because if you can do cells really well, you have a more controlled environment to kind of remove a lot of the technical hurdles to showing that the platform works. And as far as specific data on tissue, I would just say stay tuned. We'll be making some announcements specifically around that very, very soon.
I've got a quick one. What's the maximum insert size that this sequencing platform can deal with?
Yeah, so great question. I think so. I would say without changing yield, dramatically changing yield on the platform, we've gone to above 2 kb. But the real answer, the detailed answer is it depends on how tightly you can maintain the insert distribution. So if it's a broad distribution, say you're using a bead-based purification, I would say you're probably best to target 1.5 kb or so as the median of that distribution. And then you'll have a tail that'll go out to 3 kb and you'll have a tail that'll go down to maybe 800 base pairs or so. If you can do a gel size cut where you can maintain that a lot tighter, you can actually go quite a bit bigger. And there's a relationship there that there's also like other platforms that use an amplification on the flow cell, there's always a bias to the shorter size because they amplify more efficiently. So if you can avoid the short tail, then you can push much larger. Probably I would say comfortably, probably to 2-ish kb is very simple. Anything bigger than that, we start introducing some longer amplification times because those longer inserts, the signal we detect on the platform is related to how many copies are present of the insert, and the longer the insert is, the longer it takes to, of course, amplify. So you can also play you can make some recipe changes to amplify a little bit longer to keep your signal up. And the same thing if you've got a bunch of bright signal from short inserts, that affects your ability to detect the long inserts. And so you just have to, if you can strike that balance, you can push to, call it between 2 and 3 kb.
Awesome. And is a Pippin good enough as a
It
gel
is. Yeah.
base?
Yeah, it is.
Okay. Awesome.
Yeah, we use the Pippin internally quite a bit. Yeah, and that's so we have some ladders where we've done 5, 5, 3, 2, 1.5, 1, 800, and where we can actually, and we can run them in independent runs, and you can see that it's pretty robust when you use Pippin.
All right. So let me ask you one other question. So you said the hybridization is stable, sort of, for a period of days, are the thick cells.
Yes.
Is that good enough that we could actually ship these to another vendor, or is there a really strong argument to have a local instrument to avoid the shipping?
Yeah, so for the conditions we've tested, you can. So we routinely have fixed cells shipped from laboratories outside Element to Element for processing within Element, for things like cell onboarding, proof of concept testing, et cetera. So you can absolutely ship the fixed cells. Now, we also know internally, though, again, depending on the cell types, depending on the experiment, you do see some. And again, I don't have a quantitative amount to tell you. I would say the experiment run fresh on a local instrument is always a bit brighter. It's a bit better than on the shipped, but the shipped works fine. I would say there's no hesitation. I think we've seen, no difference for things that are, say, walked across campus versus shipped on dry ice or shipped ambient. But I will say on the Trinity side, the Hive capture side, those are absolutely stable frozen. The Hive is stable frozen, so there would be no issue in shipping something like that. We've tested out to 1 week at -20, and I don't know how much longer we've tested beyond that, just out of need. But we know it's absolutely stable with no change in performance, say at a week. So there'd be no limitations in shipping for the Trinity workflow.
Thanks.
Well, great. I know we're at the, just at time. Want to be respectful of everybody's day. Again, thanks for your attention on a snow day. And again, if any questions come up, Chris and I would be more than happy to answer them.
Thanks very much. Bye, everybody.
Thanks, everybody. Thank you, Shawn.
Thanks.